Hello everyone, welcome to the 1000 MCQ series of GPAT examination. This is an online coaching course exclusively designed for GPAT aspirants. So stop dreaming and start doing. Let's start with the first question of the day. Which of the following drug is used to demonstrate the topical anesthetic action on eyes? Let's see the option. Is it ketotifen, tolmetin, nifedipine or lignocaine? See, lignocaine is a local anesthetic. These are used to numb the surfaces of eye in many conditions such as for cataract surgeries and other medical conditions. Okay, coming to the other option, ketotifen. Ketotifen is an antihistamine. Antihistamine means it will be mainly used for allergic reaction. It is a mast cell stabilizer. Okay, it is used for allergic conjunctivitis and these are administered as eye drops. Okay, coming to tolmetin. Tolmetin is an NSAID. NSAIDs are used for pain and inflammation. These are applied topically as well as orally. Now, these are mainly used for skin conditions or skin inflammation and pain. Coming to the next option, that is nifedipine. Nifedipine is a well-known calcium channel blocker. It is used to treat high BP, that is high blood pressure as well as angina. And how are we administering it? We are administering it by orally. Okay, so coming to the question, which of the following drug is used to demonstrate topical anesthetic action on eye? So what will the answer? It will be lignocaine. Coming to the next question, glass transition temperature is detected through X-ray diffractometry, solution calorimetry, differential scanning calorimetry or thermogravimetric analysis. Here, what is glass transition temperature? The particular temperature at which a rigid or a glassy state is transferred or a transition takes place to a more flexible or a rubbery state. That particular temperature is known as glass transition temperature, from a glassy state to a rubbery state. Now, what is this DSC? Differential scanning calorimetry. It is mainly a technique where we are measuring the heat flow. Okay, heat flow into the sample and outside the sample. That is how much heat is absorbed by the sample and how much heat is expelled from the sample. That is the DSC, differential scanning calorimetry. So here, when a material undergoes glass transition, when a material changes from glassy state to rubbery state, there is a change in the heat capacity, which can be detected by using the differential scanning calorimetry. Right. Coming to the other techniques that were discussed in the option, X-ray diffractometry. It is mainly used to understand or study about the crystal structures. Solution calorimetry. Solution calorimetry measures the heat of the solution when a substance is dissolved in that solvent. Okay, that is solution calorimetry. Thermogravimetric analysis. Thermo, heat, gravimetry means weight. That detect the change in the weight. That can be due to decomposition or any other type of process. Right. So these are the analytical tools. Now, glass transition temperature can be detected through what? Through differential scanning calorimetry or DSC. Now, coming to the next question, collagen is an example of, is it a polypeptide, dipeptide, tripeptide or oligopeptide? What is a polypeptide? Polypeptide means it is a chain of multiple amino acid which are linked together by peptide bond. Dipeptide means two amino acid. Tripeptide means three amino acid. Oligopeptide means these are short chain amino acid sequences. Typically, they can range from 10 to 20 amino acid. See, collagen is a very large, complex protein molecule. It is composed of many amino acids and are linked in a specific sequence. Okay, which means collagen will belong to which category? It will belong to polypeptide category because it contains many amino acids. Right. So, what will the answer? Collagen is an example of polypeptide. Coming to the next question. All are the names given to the molecular mechanism of skeletal muscle contraction except, okay, walk along theory, fluid mosaic theory, Ratchet's theory or sliding filament theory. See, let's see each theory in detail. First one is walk along theory. In walk along theory, what are we saying? We are telling that myosin head walks along the actin filament. Because of this, there will be a pulling, and this pulling will result in muscle contraction. What about ratchet theory? In ratchet theory, the myosin head will act like ratchets. Right? It will act like ratchet, and it will pull the actin filament in a stepwise manner. Now, what is the sliding filament theory? Sliding filament theory, there are two filaments. That is the thick filament as well as the thin filament. So, this will slide along each other, as past each other, and this will lead to the shortening of the muscle. That is described as muscle contraction. While in fluid mosaic theory, this fluid mosaic theory explains about the structure of the cell membrane. Right. Now, so what will the answer? All the names are given to the molecular mechanism of skeletal muscle contraction except for fluid mosaic theory. It is a theory for cell membrane. Now, coming to the last question of the day. The Anderson pipette is commonly used for determining particle size distribution using which method? Is it sieving method, sedimentation method, microscopy method, or all of the above? Let's see. These all are particle size distribution methods. Let's start with the first one, the sieving method. In sieving method, what we are doing? We are using different grades of sieves, and then we are analyzing the particle distribution. Right. Now, next comes the sedimentation method. In sedimentation method, we are using the principle of particle settling. On what basis we are doing that? We are based on the size as well as the density. Here, we are using Anderson pipette in sedimentation method. Okay. Next comes laser diffraction method. This is also known as static light scattering or SLS method. Here we are measuring the angle as well as the intensity of light scattered by that particular particle. Next one is DLS, that is the dynamic light scattering. In dynamic light scattering, it will be in motion. Here we are analyzing the light scattered by the particle as such. Here in dynamic, we will be measuring the light scatter, the fluctuation in the light scattering due to the Brownian motion of the particle. The particle will be in Brownian motion, or that is the zigzag motion. Now, dynamic image analysis. In dynamic image analysis or DIA, we are capturing the images of that particle in motion, and then we are analyzing this particular image. Next comes the electron microscopy. In electron microscopy, we are using different types of microscopy such as the SEM, scanning electron microscopy, TEM, that is the transmission electron microscopy, etc. These are mainly high resolution electron microscopy. These can be used for particle size analysis, but we are mainly using it for what? So as to study the size as well as the morphology. The surface morphology also will be easily studied by using electron microscopy. So these are the various particle size distribution analysis methods. So here in Anderson pipette will be used for which method? For sedimentation method. So this is our exclusive course for the GPAT online coaching. So as to get a free demo live class, you can fill out the Google form link given in the description box. Hope you liked the video. Thank you so much.